So one part is normalization, but then we also want to select genes to work on. If we work on the full gene space, there's quite a lot of noise. So we want to get the genes, basically just get an improved signal to noise ratio, and also to minimize the data set. So if we select a few thousand genes to do PCA and everything on, it goes so much faster. And the most common way is to just select highly variable genes. But there are some other methods, like taking top PCA loadings or correlated gene pairs and so on. I'm not going to go into those. So what are the variable genes? And the whole idea is that we should have genes that have a mean variance higher than what we expect by random. And one common way is to calculate the coefficient of variation. Another way is to estimate high dropout genes. So I think this was pretty much the first paper published on this idea of detecting highly variable genes, where they use spike-ins. So it's plotting the mean expression versus coefficient of variation in squared. And then they fit a line to all these blue dots that are the spike-ins. And then estimate based on the dispersion of the spike-ins, a second line above that one, where they say this is the cutoff for technical variation. Anything that's within this region, we can't say for sure if it's technical or biological. But if it varies more than the spike-in, we can say that these are most likely biological variation. However, in quite a lot of your data sets, you will not have spike-ins, but you do sort of the same assumption that most genes are not biologically variable. So you just take all genes in here, you fit a line, and then you define a p-value cutoff for how far above this line do we draw the cutoff, and then select the top variable genes. And the exact way of estimating this dispersion or variation will differ a little bit from package to package, and there's several different methods for it, but they're all very similar, I would say. And especially if you have spiking controls, you need to realize that what normalization you do has a big impact on how you define your variable genes. It's almost the case that you have to do a length normalization if you use spike-ins, because the spike-ins are short compared to normal genes, so they will have fewer counts, and that whole curve will be shifted down, and you will overestimate the variable gene selection. Then there's another method, sorry, called M3 drop. They just have a slight different way of modeling. It sounds super fancy. They're doing this Michaelis-Menten equation about dropouts, but it's actually been shown in some of these evaluation papers that this one doesn't give as good results as these normal variable gene selection methods.